Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I am Professor Dr. Shehzad Farouk. Growth hormone is synthesized in somatotroph cells, which are a subclass of pituitary acidophilic cells. The concentration of growth hormone in the pituitary is 5 to 15 milligram per gram, which is a much higher than the microgram per gram quantities of other pituitary hormones. Means the concentration of growth hormone is in milligram per gram, while the concentration of other pituitary hormone is microgram per gram. So the growth hormone is in higher concentration. Growth hormone is a single polypeptide with a molecular mass of about 22 kilodalton in all mammalian species. Its gene is located on chromosome 17 and it is water soluble. Growth hormone has 191 amino acids and its half-life is 20 to 50 minutes, and this is the most abundant and human growth hormone made by recombinant DNA technique is available for therapeutic use. This is the signal transduction pathways activated by the interaction of growth hormone with its receptor. The growth hormone receptor is a member of cytokine hematopoietin receptor superfamily. The growth hormone receptor is a protein with molecular mass of about 70 kilodalton with a single membrane spanning domain. Growth hormone binding causes the dimerization of two growth hormone receptors. This results in the activation of growth hormone receptor associated JAK2 tyrosine kinase and phosphorylation of the receptor and JAK2 on tyrosyl residues. These events results in the activation of number of signaling pathways, including STAT protein phosphorylation and gene transcription. The second pathway is SHC and growth factor receptor bound protein 2. And this lead to the activation of MAP kinase pathway. Then comes the IRS phosphorylation with activation of phosphatidyl inositol 3 kinase. And lastly, phospholipase activation with the production of diacylglycerol and the activation of the protein kinase C. These four pathways are activated mainly. The JAK kinase pathway is unique to this class of receptor since the other pathways are activated by number of different hormone receptors. These are the structures of growth hormone releasing hormone and growth hormone release inhibiting hormone. There is a disulfide linkage in growth hormone release inhibiting hormone. There is no such disulfide linkage in growth hormone releasing hormone. The control of secretions of growth hormone, as it's clear from this diagram, that exercise, stress, decrease blood glucose level, increase blood amino acid level, and the sleep rhythm causes effect on hypothalamus, and they cause increase production of somatostatin, which is a growth hormone release inhibiting hormone, and inhibitory effect on growth hormone releasing hormone. Resultantly, the activation of somatostatin causes decrease activity of anterior pituitary to produce growth hormone, and same results from the inhibition of growth hormone releasing hormone inhibition. The growth hormone produced by the anterior pituitary, when the growth hormone releasing hormone activated in other conditions, in the reverse conditions, causes production of growth hormones, which when affect on the liver causes production of IGF-1, insulin-like growth factor 1, and the production or the concentration of IGF-1 causes inhibitory effect on growth hormone production from the anterior pituitary, and it also act on the hypothalamus to inhibit the production of growth hormone releasing hormone. Physiological and biochemical action. Growth hormone is essential for postnatal growth, carbohydrate metabolism, lipid metabolism, nitrogen metabolism, and mineral metabolism. The growth related effects are primarily mediated by IGF-1. Effects of growth hormone on protein metabolism. In general, the growth hormone stimulates protein anabolism in many tissues. This effects reflects as increased amino acid uptake, increased protein synthesis, and decreased oxidation of proteins. The effects of growth hormone on carbohydrate metabolism. Growth hormone generally antagonize the effect of insulin. Hyperglycemia after growth hormone administration is the combined result of decreased peripheral utilization of glucose and increased hepatic production via gluconeogenesis. In liver, growth hormone increases liver glycogen, probably from the activation of gluconeogenesis from amino acids. It causes impairment of glycolysis at several steps and the mobilization of fatty acids from triacylglycerol stores may also contribute to the inhibition of glycolysis in muscles. Prolonged administration of growth hormone may result in diabetes mellitus. Lipid metabolism. Growth hormone promotes the release of free fatty acid and glycerol from adipose tissues and increase circulating free fatty acids, and this results in increased oxidation of free fatty acid in the liver. Under conditions of insulin deficiency, that is diabetes mellitus, there is increased ketogenesis may result. These affect and those on carbohydrate metabolism probably are not mediated by IGF-1. Effects of growth hormone on mineral metabolism. Growth hormones, or more likely IGF-1, promotes a positive calcium, magnesium, and phosphate balance, and it also causes the retention of sodium, potassium, and chloride. The first effect probably relates to the action of growth hormone in bones, where it promotes growth of long bones at the epiphyseal plate in growing children and appositional or acral growth in adults. In children, growth hormone also increases formation of cartilage. Prolactin-like effects of growth hormone. Growth hormone binds to lactogenic receptors and thus has many of the properties of prolactin, such as stimulation of mammary glands and lactogenesis. Now, pathophysiology, or the disorder associated with growth hormone. Hypopituitarism, there is decreased secretion of growth hormone, whether from isolated growth hormone deficiency or panhypopituitarism, are the most serious in infancy because affected infants fail to grow properly. The other metabolic effects are less troublesome. Several types of dwarfism help illustrate the importance of various steps in growth hormone action. Growth hormone deficient dwarfs respond normally to the exogenous growth hormone. Two type of target organ resistance have been described. Laron type dwarfs have excessive amount of growth hormones, but they lack functional hepatic growth hormone receptors. Pygmies apparently have post-growth hormone receptor defect, and this may be limited to the action of growth hormone, which exerts through IGF-1. Dwarfism, that is childhood and hypopituitarism. This is due to deficiency of anterior pituitary secretions during childhood. Parts of the body develop in appropriate proportion to one another, but rate of development is decreased. A patient of hypopituitarism, dwarfism, not secrete sufficient quantity of gonadotrophic hormone to develop adult sexual function. Treatment with human growth hormone replacement is indicated. Right. In this slide, you can see the relationship of growth hormone IGF-1, IGF-2 to dwarfism. You can see the growth hormone deficient dwarfs only respond to the administration of growth hormone. They have low growth hormone level, low IGF-1 and IGF-2 level. Pygmies have normal growth hormone level, low IGF-1 level and normal IGF-2, and they do not respond to growth hormone stimulation. Pygmies apparently have post-growth hormone receptor defect. In Laron type dwarfs, there is high growth hormone level and low IGF and IG2, and this group also do not respond to the growth hormone because it lack functional hepatic growth hormone receptor. In this case, there is no functional hepatic growth hormone receptor. Excessive secretion of growth hormone, gigantism and acromegaly. Gigantism results from the excessive release of growth hormone if it occurs before the epiphyseal plate closure, since there is accelerated growth of long bone. And acromegaly results from excessive release of growth hormone that begins after epiphyseal closure and the cessation of long bone growth. In gigantism, it occurs before the epiphyseal plate close, and in acromegaly, it begin after the epiphyseal closure. So, the symptoms differ in these two groups. In both groups, there is excessive release of growth hormone, but presentation differ due to the disease occurring after the epiphyseal closure or before the epiphyseal closure. The gigantism, the acidophilic growth hormone producing cells of anterior pituitary becomes excessively active and large quantities of growth hormones are produced. If this condition occur before fusion of epiphyses of long bones with shaft, there is highest increase in height so that person become a giant, that is 8 feet approximately. Hyperglycemia due to insulin producing beta cells of pancreas exhaustion. To overcome this hyperglycemia may result. This may lead to diabetes mellitus. If remain untreated, microsurgical removal of tumor or treatment by irradiation of the pituitary gland is indicated, but if it remain untreated, death in early childhood may occur. In case of acromegaly, if the acidophilic tumor occur after adolescence, that is after fusion of epiphyses of long bones with shaft, person cannot grow taller but becomes thicker. Soft tissue can continue to grow. Enlargement is more in bones of hand and feet and membranous bones. Acral bone growth causes the characteristics facial changes. Lower jaw protrude forward, forehead slants forward, nose increases to twice the normal size. Feet require 13 size shoes and finger thickened so that hands become twice the normal size. Changes in vertebra causes hunched back known as kyphosis. About 40% of persons with acromegaly have a G protein linked disease. Such persons have one of the two mutations in the alpha stimulatory subunit that abolishes the intrinsic GTPase activity in the protein. One mutation at arginine position 201 affects the site that is ADP ribosylated by cholera toxin. Since alpha stimulatory with such a mutation is constitutively active, cyclic AMP is overproduced. This results in excessive production and release of growth hormone and in under and there is unrestrained growth may occur in this case. In this scene, the alpha stimulatory is an oncogene. This was all about growth hormones. Thank you very much, dear students. Allah Hafiz.